This summer, our team was able to publish our manuscript in Science magazine. This publication goes deep into explaining how our discovery engine works and how we were able to find many more targets accessible to molecular glue degraders, or MGDs. Today, I'm excited to be talking to two of our leading scientists, Pablo Gainza and Georg Petzold, going deeper into the manuscript and the rationale for why we published that. Georg, tell me a little bit more about the rationale.
So as you know, we are a company that's focused on molecular glue degraders, and this is this really exciting new modality that allows us to drug proteins that were previously perceived undruggable. And the reason why this is working is that the mode of action of these compounds is completely different to what was known before, in that it just glues two proteins together and then one happens to be degraded within the cell, completely removing the activity of that protein, right? So this does not require any druggable pockets on the target. Hence, there is this huge promise that undruggable proteins could finally be reached with that modality. So the parent molecule of this modality is lenalidomide, which is already used in the clinic in multiple myeloma treatment for a number of years now, more than a decade. But for a very long time, its mode of action wasn't really known, right? And it was only discovered in hindsight that it actually works through this new mechanism. And because of this more or less serendipitous discovery of this mode of action, there was a lot of skepticism around this modality, right? Many people believe that it could never be, that this is a curiosity, it could never be rationally designed. There might be, like, the early molecules were not very selective, etc., right? So there was a lot of skepticism around this modality. But we thought slightly differently. We kind of focused on this promise of reaching undruggable targets. So we really took, like, the few knowns in this modality to really understand how the mechanism is really working, in order to build predictive models and basically shift this entire field towards more rational approaches.
Okay, that's really exciting. Now, Pablo, give me a little bit more insight as to how this was actually done.
So we took this notion of distilling the knowledge that was already available about cereblon, and we used that to build two templates for the requirements to bind to the cereblon target with molecular glues. One was a more common structural template, and the other one was a surface template, where we looked at proteins purely from the perspective of their surface and not really what's underneath them. And then we used artificial intelligence machine learning methods from the field of geometric deep learning to project this across the proteome and find other proteins that could also be amenable for the modality. Once we established our map, we used an array of complementary techniques from proteomics, structural biology, and different cellular and biochemical assays to actually validate this. And what's really interesting about this is all these learnings actually feed back into our models, help us improve them, and can actually help us further expand this target space.
Okay, this is really amazing. Now, and I know there are many paradigms that were broken, but tell me, what does this mean for the field of molecular glue degraders?
Well, I mean, when we started the work, we only knew about, like, roughly a handful of targets, right? And through our mining approaches, we have a much more complete understanding of the CRBN target space now, and it is clear that many more targets, many more proteins can be reached by this modality. So we did use rational approaches to expand the CRBN target space, number one, right? We could also show in the paper that molecules actually can be designed that are highly selective against individual targets, which was also a perception that basically kept the people away from this modality.
One of the things that I think is interesting is that we are shifting the field away from thinking about diagrams as these sequence motifs or structural motifs and thinking them more about the surfaces and the complementarity that they have with something like CRBN. And the broader implications for the drug discovery field is that really we could go after many more targets than those that we have discovered so far, thanks to this reprogrammability. And the other broader implication is that we can actually go about it rationally, and we can use AI and machine learning tools to broaden the space of cerebral targets.
This is fantastic. Thank you very much, both of you. And I'm really excited to see what's coming next.